So what's the benefit of having this supercoiled structure of your DNA? Think about it. I mean, the long DNA strand, if you want to contain that in a small space called nucleus, you want to condense it as much as possible, right? So this is more condensed storage form of your DNA. So you can package a lot of material into a very small space. Okay, so it's packaged conformation. Okay, but that packaged conformation could be a problem when you come to a time when you need to replicate your DNA, because the enzymes should be able to find the place where they need to start the replication or transcription. Okay, so under those conditions, you have to sometimes relax these supercoiled conditions to a kind of relaxed form, so the enzymes can come in and bind with DNA and do their job. So for that, so of course there are enzymes called helicases that will relax those DNA strands. But when helicases relax those DNA, then one side may be relaxed, but the other side may not be relaxed. Actually, that will be more compressed. Okay, so during the DNA replication process, there are these special enzymes called topoisomerases that will actually relieve the tension or the supercoiling that is created by helicase enzymes. Okay, so there are topoisomerase 1, topoisomerase 2. They are the actual enzymes that can relieve the supercoiling by cutting the DNA, okay, running and the other strand, uncut strand through that cut strand. Okay, by doing so, they can relieve the tension, the supercoiling. And topoisomerase 1 will make single cut, and topoisomerase 2 will make double cut, and both of the enzymes will deal with the supercoiling problem within your cell. Okay, so topoisomerase also decantate, meaning after the end of the replication, two replicate DNA should be separated out. That's what decantation means. Okay, so maybe replicate DNA should be separated out. That's what decantation is. Okay, or unknotting. Okay, separating out, and distangling, so something like that can be distangled this. So those are all managed by these unique enzymes called topoisomerase. Okay, how do they do their job? So this enzyme is a protein, of course. It can bind with the DNA, okay, and then once they bind, it can break the phosphodiester bond. Okay, and then there are subunits doing different jobs within that enzyme, but type IB mediates a controlling rotation mechanism to relax both negative and positive supercoils. By IB cleaves the single strand, like I said, of the duplex DNA, and attack the opposite side with the tyrosine on the DNA. It's got tyrosine amino acid and attacks it to yield a 3' linkage of phosphotyrosine intermediate with 5' OH group. So that's what it means. The tyrosine is there, so that's the intermediate. Okay. All right. Anyway, very interesting enzyme. All right. So again, showing you the process of topoisomerase handling the supercoiling problem. And then you can actually visualize the change of DNA conformation from maybe supercoiled structure to a little more relaxed supercoiled, and then maybe mixed circular, and then linear. Okay, especially if you're dealing with bacterial DNA, which is just a circular form. But if you isolate these circular chromosomal DNA from bacterial cells, many of them is isolated in the supercoiled form like this. So the isolated DNA, if they are separated out, agarose gel electrophoresis, okay, without enzyme cutting, mostly they may come in as maybe supercoiled form like this, or a little more relaxed supercoiled form, depends on how much of the supercoiling happened. Or they could be totally relaxed as the circular form like this as well, if there was any nicking happened, like the work of topoisomerase. But nicking could happen through your pipetting or vigorous physical treatments. Okay, so they may exist as tightly supercoiled or loosely supercoiled or circular. We call the nick circular conformation. Depends on what conformation they take up. How much they migrate within the gel would be different. Because this is very condensed form, they take up very little space, so they can migrate much faster within a given space of the gel and concentration in a given time. This one will migrate, slower than this, but migrate faster than that. And the Nick circular, since it's very widely, taking a lot of space, it's going to migrate much slower. Okay. If both of DNA strands are cut, of course, the circular form will become linearized like this. Okay, and the linear conformation will migrate faster than circular, but migrate slower than these supercoiled. So actually, you can see maybe 4 different conformations of the DNA showing up on the electrophoresis agar gel: supercoiled, tightly, and loosely supercoiled, Nick circular, and even linear form. Although they are all the same DNA, chromosomal DNA. Okay, so it's the topological difference of the DNA. That's what it is, the dimensional structural difference. And when you do electrophoresis, gel electrophoresis like that, the DNA bands will not be visual unless you use a dye molecule that can bind on the DNA, give you a signal, visible signal. Okay, so normally a dye molecule we use is ethylene bromide. Ethylene bromide can bind with your bases of DNA very well. So ethylene bromide looks like that. It has a unique structure. It's a fluoropore. It receives fluorescent energy, and then that fluorescent energy is emitted from them. So you can see kind of bright orange color if ethylene bromide binds with the DNA from DNA molecules when you shine the UV light on. Okay, so anyway, so ethylene bromide goes between the bases, and they bind with them, and they don't come up easily. So it binds with DNA well. Once they bind, they don't come up. So if DNA is used for DNA replication, there may be mutation happening. So we call that ethylene bromide is a strong mutagen, can change the DNA sequences. But anyway, the important thing is ethylene ion binds to DNA, cause it to unwind because it binds with so many bases. Okay, so you can make supercoil into maybe relaxed form. Depends on how many etherium is binding among the bases. That's what you are seeing there. Okay, you can make it unwind, supercoil form into maybe circular form. Okay. So, after understanding the structure of DNA, you may wonder, okay, so DNA molecules adapt that double strand structure. 2 strands come together through hydrogen bonding, right? Why would they adapt double strand structure? I mean, just want to think about it. Can they just use a single strand DNA? What would be the benefit of adapting the double strand structure? Okay, and so if you think about it, if you have 2 strands, of course, you can store more information. Most information storage is possible. That's a benefit, right? Yes, and another thing is, by having 2 strands coming together, okay, and hiding bases in the middle of it, they can provide more protective structure because bases would be protected from outside. Okay, they can protect the integrity of the bases, and then facilitate replication. Okay, so both strands can be replicate at the same time. So those would be benefits of having the double strand structure for DNA molecules. Okay, but we talked about this lecture being talking about nucleic acid, right? Nucleic acid is not all DNA. There are different nucleic acids. Those are called RNA, ribonucleic acid. Okay, there are a whole bunch of ribonucleic acid within the cells. There are a whole bunch of them, actually more than DNA. Okay, so now you need to understand what RNA molecules are, ribonucleic acid, okay, how they are different from DNA. So number 1, RNA contains ribose, okay, like DNA, but that it's a ribose, not deoxyribose, okay, meaning there's no deoxy, so there's OH there, there's OH there, so there are double OHs in this sugar, okay, that's the difference. Instead of deoxyribose, it's the ribose. And then it contains uracil, okay, instead of thymine. So, see, uracil is that, but DNA contains thymine, right? So there's minor structural difference, but RNA contains uracil. Okay? And then another important thing, RNA molecules exist as single strand instead of double strand. Okay? RNA is a single stranded nucleic acid. Okay? So those are 3 unique entities of RNA molecules. Ribose instead of deoxyribose, they have U base instead of T, and normally they use a single-stranded conformation. Okay, then would they exist as something like that? The answer is no. They will form maybe a loop or hairpin structure, like maybe something like this. This is maybe a loop or hairpin. Okay, so that's what they adapt. So by doing so, they can protect their base integrity a certain degree. Okay, and there's a question. So we said RNA has uracil instead of thymine, right? So there's a little difference. Okay, what would the benefit of having uracil or thymine instead of uracil if you are DNA? So most of the living cells adapt DNA as the main genetic materials, right? So maybe think that DNA may be a better form, okay, better nucleic acid to use as a genetic material, right? So then if you said that, then what would be the benefit of using T over U, okay, in your DNA molecules? Think about it. Okay, to answer that question, you need to understand, the cytosine, okay, can become uracil. Okay, so what's the structural difference between these two? So if this is removed, you can make this, right? So we call it deamination. So amino group is removed, right? So if cytosine is deaminated, if you chop that up, that will become uracil. Okay, so cytosine is normally found from DNA and RNA or both, right? But think about DNA molecules having this conversion. Okay, so if you have U's through cytosine deamination, you can easily recognize that because DNA molecules do not have U normally. So if you see U's, that means your cytosine was deaminated. So that's a mutation, right? So then if you have an enzyme that can recognize this change, uracil, and then they can maybe fix it back to cytosine. Okay, so yes, the cytosine deamination can be easily recognized if you don't use uracil in your makeup and the DNA molecule. That could be a main advantage of using thymine instead of uracil in your nucleotide. Okay, make sense? And then another thing is, like I said, RNA doesn't exist as a relaxed single strand. Instead, it folds up. Okay, make these, hairpin structure, or maybe kind of stretch the loop structure. Okay, how is it possible? It is possible because the nucleotide bases within its single strand structure can also make hydrogen bonds, okay, among themselves. So if you make these base matches within this area, stretch area, you can make this hairpin structure. It looks like a hairpin, right? So that's a hairpin, and the entire thing may be considered as a loop. Okay, so maybe bulging out, in the middle of the loop. So RNA adapts this complex structure as well. So that's how they protect the integrity of the bases. Okay, so I told you about the looping and hairpin formation of the RNA molecules. So since RNA molecules will allow kind of a non-Watson and Crick base pairing, what it means is C to G and T to A base pairing. Those are normal Watson and Crick base pairing rules, right? But under certain conditions, RNA will allow odd base pairing. We call that non-Watson and Crick base pairing, like between G and U. G and U normally do not pair up. Instead of normally it should be G and C, and maybe U and A. That's the correct base pairing. But this odd base pairing may be possible within RNA molecules. Since this odd base pairing is possible, they can make more of these loop structure, hairpin structure, and that's the way of their protection. They're protecting their base integrity. They do that. Okay, and double helical RNA adapts a structure close to A form. We already talked about it. If it's RNA and RNA double strand, it's going to form an A form, which is kind of fatter, double strand, coil without major and minor group. So we understood that, right? And then this is kind of unique RNA structure change that allows the gene regulation. So gene regulation means your gene is turned on meaning transcription, translation is happening, so the end product, maybe protein is made a lot. That's what gene expression, right? But that process can be regulated, maybe changed out by some